When we look at the brain, once again, you can look at specific structures and begin learning how to recognize them by identity or their appearance. But once again, to be able to put it into some kind of system that will translate to different modalities, we have to be a little bit more systematic. And the way we do that is I will make reference to a mid-sagittal slice right over here, as we looked at earlier, and I'm going to break it into 7 divisions. Excuse me. As we look at these divisions, you're going to find there are key structures that are found in what I'm going to define as the upper cerebrum, key landmarks found in the division of the cerebrum. Basal ganglia are specialized gray matter structures that help us in terms of interpretation, also as relay centers of impulses from one portion to the brain. At this level or division, you're going to find there's a key collection of key landmarks. Midbrain is another one that's going to be rich in structures and landmarks, and will identify those structures at a given level as you work down the brain, the pons, cerebellum, medulla. So when I teach this course to our students, rather than give them a global perspective, we start from the top, we work down toward the lower portion of the medulla, and we identify key landmarks as we go through. And again, the system seems to work well in terms of students being able to translate that knowledge into different portions of the brain and other modalities. The cerebrum is really the area of higher intelligence. We have a lot of problem-solving skills. Higher order learning occurs in it. There are 5 lobes that make it up. We'll be seeing primarily the frontal, the parietal, to a smaller degree the occipital, and then you get down into the lower portion, referred to as the temporal lobes. For many early anatomists, they oftentimes would refer to the cerebrum as having 4 lobes. In reality, there was another one that was really responsible for different functions, mostly in terms of learning, deeper learning, going from short-term to long-term memory. And it's referred to as the insula, or the hidden lobe. In some textbooks, they call it the central lobe. And we're going to find it as just a small area of gray. Easy to understand how early anatomists couldn't find it because it's such a small area. But as you look at the cerebrum, it's going to be made up of 5 lobes. So we'll look at some of these as we work our way down. Here, this whole area right through here is all cerebrum. And of course, we're getting a different perspective. We have basal ganglia and other structures here. But it gives you an idea of the massiveness of the cerebrum. Once again, in this line drawing over here, you see that little corpus callosum that's going to link one portion of the cerebrum to another. I want to get into dural coverings as well, and that's kind of a term I give. There's a series of tissues that cover both the brain and spinal cord that are called collectively the meninges. The toughest, the most durable one is referred to as the dura mater. And the dura mater basically is the outermost layer, and it's going to be found covering the brain, but more importantly, there is a dividing point within the fissure that separates both hemispheres of the cerebrum. And within it, you're going to have a double layer of dura mater that we give it a term falx cerebri. And the falx cerebri becomes a very important landmark for a couple of reasons. First of all, it will present itself on T1 weighted MR, T2 to a certain degree, certainly on CT when the patient's been given IV contrast. So it will serve as a landmark that we can identify directly down the longitudinal fissure. But what really makes it important is that you have certain regions or folds. If you were to go through a cadaver brain, you would find like a small tunnel or passageway within the upper portion of the falx cerebri. What goes through it is a large volume of blood that we call in this region a sinus. And the superior sagittal sinus is a massive collection of blood that runs from anterior to posterior. And it's going to be seen as, well, depending on the type of pulse sequence you do with MR, either as a flow void artifact, or in some cases as a large density running right down the middle of the longitudinal fissure. it's contained within the upper portions of the falx cerebri. It's interesting because when you look at venous sinuses, all this is going to be venous blood. Eventually, it's going to be skirted through the brain and brought through jugular foramen, and those then become internal jugular veins. The superior sagittal sinus is interesting because our sinuses in general, because they have two features, or they're absent of two features, I should say, that make them different from veins. First of all, there's no vessel wall that contains that venous blood. Basically, it's the confines of the upper portion of the falx cerebri that keeps that blood intact. Secondly, basically, there's no valves. All veins basically have valves that help bring it back to the heart, to the right side of the heart. These basically are going to be without valves, and what's going to drive the blood at an extremely fast pace is intracranial pressure. These will show up as important landmarks from that standpoint. There's a smaller one right over here called the inferior sagittal sinus. It's going to be smaller in size. It doesn't really show up as a standard landmark. Eventually, it's going to terminate in one called the straight sinus, and then they're all going to communicate at one point, which we're going to call the confluence of sinuses later on. This superior sagittal sinus will stand out as a landmark, and as you work your way down the cerebrum, down into lower portions, you'll see it as somewhat of a triangular-shaped defect or flow void artifact that will serve as an important landmark. We'll point it as we work our way down. Here, we're going to look at the upper cerebrum, and you're going to find that there are very few landmarks that we have to worry about. Here's that longitudinal fissure that basically separates both hemispheres of the cerebrum, and within it is going to be the falx cerebri. So here's the thing that we have to keep in mind. When I point to this area, when I'm working with my students, it could be one of three things. It could be a portion of the falx cerebri, it could be a portion of the superior sagittal sinus, or the longitudinal fissure itself. What you see on the direct posterior midline is where that superior sagittal sinus is beginning to work its way down posterior and inferior, and it gives you somewhat of a triangular-shaped defect along that portion. In the upper cerebrum, the other things we're going to point out is a massive collection of white matter tracts that basically, if you were to look at the spinal cord, you're going to have all these tracts come up into the brainstem. There's going to be a point where the motor and sensory fibers from the right side of the body are going to cross to the left. We'll talk about that. That occurs in the medulla. But the point here is that you're going to have this large burst of white matter tracts that then are going to be skirted off into different portions of the brain. This area we're going to call the centrum semiovalle. It's perhaps the most dominant feature within the upper cerebrum. If you were to have a stroke or injury or trauma in this area, one half of the body basically would lose both motor and sensory function. So that would be a real important landmark that we're going to emphasize. As we work into the cerebrum itself, certain things we've talked about before, this would be anterior, this would be posterior. We'll find out this is the genu. Back over here is going to be the splenium. We'll talk about the ventricles, we'll talk about this little membrane, and then we'll see the first of several basal ganglia. So we start getting introduced to basal ganglia in the cerebrum area. And again, we look at CT, but we won't focus on that. As we work into the basal ganglia, we're going to give names and terms for these different gray matter structures. We'll call these caudate nucleus, the lentiform nucleus. We'll talk more about that as we work our way down. Again, I'm going to scoot through the CTs. Here, we'll get into the lower portion of the basal ganglia. You'll still get a glimpse of certain structures right through here, but also you're going to pick up structures called the thalamus, which are large paired gray matter structures that help us in interpretation of different impulses to from the brain, from the white matter tracts, and it will skirt these impulses in different portions of the brain. These thalamus are going to form the lateral wall of the third ventricle. Midbrain, my favorite area or level. Excuse me again. The Mickey Mouse sign, we're going to find out that the cerebrum basically is going to connect at least the cerebral cortex from the upper portion of the brain to the brainstem through these things which we call peduncles. We're going to find out that there's a lot of key anatomy in this portion of the midbrain. We'll talk about the substantia. We'll talk about cerebral aqueduct. Here's another connector, which would be the vermis. And then back over here, which I didn't really emphasize before, is a layer of dura mater that is found along the posterior portion of the brain that separates the cerebrum from the cerebellum. We call it tentorium cerebelli. Early anatomists thought it was in the shape of a tent. In fact, when you look at it on a coronal slice, it has domed tent-like appearance about it. And this little structure back here is going to be, in part, the tentorium cerebelli. So that will become an important feature as we work our way down. Pons is going to carry a number of different structures within. We see an expansion of vermis. We see the pons, which is a bulbous structure, loosely defined as the bridge. We're going to find right here is a portion of the sphenoid bone running down inferior to form a portion of the foramen magnum. And right here between the pons and the clivus is what the term is for this little bony ridge. We see the basilar artery, and this will either show up as a flow void artifact or in some pulse sequences as a little dot or density or brightness in this area. Back over here on the posterior portion of the pons, we'll pick up the fourth ventricle. We'll talk more about it here in a moment. And when you get back over here, we'll get into venous sinuses. This is where you have a collection of all the venous sinuses coming together that then will be skirted bilaterally to different portions of the lower part of the cranium and brain, where they come together and you see rather than a triangular-shaped defect, but more of a confused area. This is referred to as the confluence of sinuses, and we'll give you more detail on that as we work our way down from that standpoint. Over here is the lower portion of the brain. Here's the posterior or hindbrain. This would be the cerebellar hemispheres. The medulla is going to be seen right over here, and then we're going to work our way down into different portions from that standpoint. So that kind of gives you an idea of what divisions we're going to look at and how we're going to approach it. Let me begin